Good morning. We're going to take a look today at the different mutations that can occur when the when something goes wrong during protein synthesis. The actual definition of a genetic mutation is when the sequence of bases in an individual's DNA is permanently changed. So these mutations can be completely spontaneous, or they could be caused by an environmental factor, which we tend to call a mutagen. Mutations can occur in all types of organisms, so not just people, but plants as well, animals. So a couple of different types of mutagens can include radiation or chemicals, which you should be kind of familiar with. These particular mutagens tend to cause cancer in humans. So overexposure to radiation or exposure to certain chemicals or other types of chemicals like asbestos. So there's two different types of genetic mutations that we're going to talk about. First being germline mutations, which will occur in your sex cells or your gametes, and these can be passed on to future generations. There's also somatic mutations that can occur. These are going to be in your body cells. That might occur when your body's either growing or trying to repair an injured part, like a broken bone or something like that. So those types of mutations, the somatic mutations, tend to not cause as many problems sometimes in terms of being passed on because they're not in the line. But these are the ones, I guess both of them can lead to cancer. Because you've got some things like the breast cancer, which can be passed on through the germline mutations. But you can also develop somatic mutations from overexposure to UV light. Now, those particular types of mutations can also spread to other cells, even though they might not have been exposed to that particular mutagen. But basically, all those mutagens, what they're doing is just messing up with the sequence of bases, and then as the cells try to either replicate or go through protein synthesis, you run into problems. So we don't have a whole bunch of time left today, so we're only going to talk about this particular type of mutation, which is a point mutation. We'll talk about frameshift mutations and a couple other mutations tomorrow, or Monday. I guess it would be Monday. But what a point mutation is when you change one single nucleotide. So due to this, an incorrect amino acid could be inserted into your protein. It might not be, depending on which bases changed. Since that code is degenerate, meaning the codons, if you remember back at that protein table, the first two bases were different, but that last base could change. It would still code for the same amino acid, so that's that first option. So the mutation, even though it is technically changing your base sequence, it might still code for the same amino acid. When that happens, that's called a specific type of a point mutation, which we'll talk again about on Monday. And then sometimes the mutation, if it occurs in that first or second base, that could completely alter your protein that it's coding for. So instead of the ATA, maybe it's ATC, which becomes a stop codon. So if that was in the middle of your protein, that might cause you to have a half-formed protein. So I know that was kind of quick. It's not a lot of information, but I know you guys took the quiz and you still have the article essay to get to, which will be due on Monday also. So if you do have any questions, just make sure you holler. Have a good weekend.